Welcome. Today we are going to learn about the blotting techniques. What do you mean by the blot? Blot describes or the blotting describes the immobilization of the sample of nucleic acid on solid support, and this solid support, it may be the nylon membrane or it may be the nitrocellulose membrane. The blotted nucleic acid, it is called as the targets, and these targets now they are going to be used for the hybridization experiment. Now, the blotting techniques, they are of the three types, and these are the blotting of nucleic acids from the gels, and it consisting of the northern blot and the southern blot. Then there is the dot and the slot blotting, and then it is the colony and the black blotting. Now, going towards the overview of there is the detection. So when you look at the blotting, first you have to run the gel, then you have to transfer the bands over the nitrocellulose or the nylon membrane. Okay, if it is the DNA, it is a southern blot. If it is a RNA, it is a northern blot. Okay, next it is a dot blot. Okay, then there is the colony or the plaque lift assay. Then you are going for the pre-hybridization. In that you are going to go for the blocking, then you are going to add the labeled DNA or the RNA, that you are going to call it as and probes. Then you are allowing the hybridization. After hybridization, there are the stringency process to remove the extra probes, okay, which are unbound one, and then you are going for the detection. So this, it is the overview of the blotting. Now the typical blotting apparatus, it is of the capillary blotting, and in that here, okay, here it is an plastic tray. Okay, you can see here it is an plastic tray, and on this plastic tray, this cassette, it is going to be loaded, and this cassette, it is going to have the gel, okay, on which you have run your sample. Okay, then there is the membrane. Okay, after membrane, there are the filter paper sheets, and then there are the tissue papers, then there is a glass plate, and then there is the wet. Okay, now this, it is going to be placed over this tray, and the wicks, they are going to be added, and through these wicks there is a running of the buffer. So the tray, it is filled with the buffer, and in the buffer there is a dipping of the wicks, that is the filter papers, they are dipped, and on that filter paper this cassette of the blotting, it is going to be placed. Now, as the name suggests, capillary blotting, here with the help of the capillary action, the DNA from the gel, it is going to move over the membrane. Okay, and in this way, with the help of the capillary blotting, there is a blotting of the nucleic acids, okay, over the nylon membrane or the nitrocellulose membrane. And this, it is requiring around the overnight, that is around the 16 to 18 hours they are required for this capillary blotting procedure. To fasten the procedure, there are two methods. One, it is electroblotting, and the second one, it is a vacuum blotting. Now, in the electroblotting, the apparatus, such a type of an apparatus, it is used, okay, for the electroblotting. The same cassette, okay, the same cassette like here, okay, that is the filter papers, gel, then the membrane and the filter papers, it is going to be made, and that cassette, okay, that cassette, now it is going to be placed in this blotting unit, electroblotting unit, which has the electrodes, okay, which has the electrodes, the positive electrode and the negative electrodes, they are there, and in between that there is a placement of the cassette, and at the negative side there is a placement of the gel, and on the positive side there is a placement of the membrane, so that when there is a flow of the electric current, the DNA, which it is going to be transferred over the membrane, okay, due to the electric current which is going to flow. And now, this procedure, it is requiring around the 1.5 and 2 hours. So the procedure, it is a faster than the capillary apparatus. Now in the vacuum blotting, the vacuum, it is created, and due to that vacuum there is a transfer of the DNA over the nylon membrane or the nitrocellulose membrane, and the blotting, it is going to be done. Okay, so there are the three methods for the blotting. First one, it is a capillary blotting. Second one, it is electroblotting. Third one, it is a vacuum blotting. Okay. Now, why you have to go for the blotting? Okay, for the isolation, quantification of the specific nucleic acid sequences, and to study the organization of it. Also, you can study the intracellular localization, expression, and the regulation of the genes. Then you can also go for the diagnosis of the infectious and inherited diseases. So, with the help of the blotting procedures, okay, with the help of the blotting procedures, you can go for any one of these application. If you want to study the specific nucleic acid, you can study. If you want to know the localization of the proteins, or the you want to know the regulation and the expression of the genes, that you can also study with the help of the blotting procedures. And also you can study the infectious and the inherited diseases. I think you may have cleared about the blotting techniques. Okay, what do you mean by the blot? Okay, it is only the when you say the blot or the blotting, it is the transfer of the nucleic acid from the gels to the solid support. And here the solid support, it may be nitrocellulose or it may be the nylon membrane. Then there are the procedures like the transfer of the nucleic acids, that is the blotting of the nucleic acids in that southern and the northern blot. Then there is the dot blot, then the colony and the plaque lift assay. Okay, in this we have learned in detail about the southern blot. You may watch that video, which is previously uploaded. Now, I think you have cleared the idea about the blotting. If you like my video, please subscribe my channel and also share my video with your friends. Thank you.